Till now we finished B cell tumors. Now it's time for T cell tumors, right? So let's understand the concept behind T cell tumors. So let me make it clear first. Okay, right. So before going in detail regarding this T cell tumors and all this, so let us understand the basic concept regarding the T cell, how it is formed and what is CD4 cells, all those in a brief. I'm not going in detail because we'll deal that in microbiology. But right now, for this purpose, let's study for the study in brief. So this is bone marrow. So let's see the lineage here. As you know, stem cells, myeloid series, lymphoid series. From this you have B lymphocytes and T lymphocytes, right? These are T lymphocytes. You have got pre-T lymphocyte, pro-T lymphocyte, then you have mature T lymphocytes. These both are pre and pro-T lymphocytes are precursors, and mature T cell is mature enough. Now let's see how the precursor T cell has matured enough. Now let's see this T cell, this precursor T cell which is in the bone marrow. Let's see how it is matured. So now I took the precursor T's out. So let me draw it in a different diagram here. So right now that precursor cell has entered the thymus where it matures, right? Normally it matures in a lymphatic tissues, right? So in thymus. So let's see, not in the thymus, but these conditions you can find wherever lymphatic tissue is present. But right now let's speak the T cell is maturing in the thymus. So right now this is thymus, right? You have got a precursor T cells, right now it has entered the cortex capsule. So in capsule right now T cell is negative for CD4, CD8, right? And it's just positive for CD45 and CD25, right? Just remember that I started speaking about the T cell maturation. In T cell maturation, I started speaking about the entrance of precursor T cell to the thymus. The first step, it has entered the capsule. At capsule, it is negative CD4 negative. That means it is not differentiated. The cluster of differentiation is not seen for the 4 and 8. Later it enters inside, so it is cortex. Right now what happened? It is double positive. That means it has gained to the entrance from the capsule to the cortex. It has gained the double. It has gained the positivity, and that positivity is CD4 positive and CD8 positive. Later then it goes into the medulla. Later this CD4 converts into helper T cells and helper T2 cells. So you have got CD4 positive and CD4 positive. That means this double positive of CD4 and the CD8, right now CD8 has separated, right? Now CD4 again divides into helper T cell, right? And you have got helper T cell 1 and helper TH2, right? Later in further cases you just remember this, that the T cell lineage, the maturation of T cell from the capsule to the medulla, right now we have T cells. Right now this is helper T cells, right? CD4. And now we are speaking about a lymphoma or the tumors which is affecting these CD4 cells, right? It is affecting these CD4 cells. Hope you understood the basic concepts, but let's go in detail regarding the notes. So first it's written that it arises from CD4 positive T cells. I showed you what is CD4, right? It arises from that point of view and associated with HTLV, right? You have a virus, this HTLV, one viral infection, it is associated with this. That means you have a protein known as TAX protein which helps in the progression of tumor cells, right? Just remember that the moment this is T cells, when this virus enter it, right, it starts proliferating. It goes and disturbs the DNA, it regulates the DNA mechanism, and you can see lots of CD4 cells, CD4 cells everywhere, CD4 cells, right? And it is aggressive, right? A for adult, A for aggressive. And remember that leukemia progression occur, that means from the lymph node, right, from the lymph node, the cells which are present, CD4 positive cells, T cells, it is arising from this, right? It is arising from CD4, and you have got lots of tumor cells from the lymph node. It can enter into the blood. Once it enter into the blood, and now it is known as leukemia. So leukemia progression has occurred, right? These tumor cells can spread, it can go to cutaneous, and it can spread, and cutaneous has involved, it can go to the hepatosplenomegaly, liver, it can go to lymphatic, where there is lots of proliferation, and you can see hepatosplenomegaly, then lymphadenopathy. And remember that even these CD4 T cells, they release interleukins that also activate the osteoclastic activity, and you can see hypercalcemia, and that is paraneoplastic syndrome, right? Paraneoplastic syndrome, because of this interleukins which has spread, right, it results in hyperkalemia. It is known as paraneoplastic syndrome associated with this. Now when you see peripheral smear, right, when you see peripheral smear, when you see this in this adult T cell leukemia, the nucleus of CD4 T cells looks like flower cell, that is clover leaf cells. This is a clover leaf, the nucleus look like a clover leaf, right? I repeat once again, we started speaking about the T cells, and in that we started speaking of the adult T cells. Why it is adult T cell? Because it has matured enough. CD4 has matured enough now. That is the reason it's adult T cell. It can be, indeed goes, it may go into the blood, known as leukemia. It can be only in the lymph node, known as lymphoma. It arises from the CD4 positive T cells. It is associated with, what is it associated? It is associated with HTLV virus, one viral infection, and this is aggressive tumor, and leukemia progression occurs, and when it reaches the skin, even that also, skin also have a lymphatic tissue, even you can find a tumor over there also, right? It can be with a leukemia where it can enter directly, or it itself can show a tumor, so that is known as cutaneous involvement. Now hepatosplenomegaly is seen because even in spleen also you have lymphatic tissue. So if there is lymphatic tissue, it might be the T cells can go there, or there itself you can see the mutation of the CD4 T cells. So that is the reason you have hepatosplenomegaly. Next we have lymphadenopathy, and because of CD4 T cells which release like interleukins which acts on the osteoclast, same way one of the mechanism is that where you have increased in hypercalcemia. So that is known as paraneoplastic syndrome. It is associated with clover leaf cells. So that is all you should know regarding the peripheral T cells, that is adult T cells leukemia. So next, let's understand the basic concept of mycosis, fungoides, and Zeri syndrome. So let's understand. Let me read first from our notes, then I'll teach you in detail the concepts behind it. So I have told you the basic concept of CD4 T cells, how it has formed. Now from premature cells has entered the lymph node. From the lymph node it has this is a process I have told you before, and right now we have got CD4 cells, right? And remember that this is our skin. When I take the section of the skin, you have got epidermis and even you got dermis. A skin has a lymphatic tissue, right? A skin has a lymphatic tissue. Now what happens in mycosis fungoides is the lymphatic tissue which is present in the skin has been affected, right? In mycosis fungoides, the lymphatic tissue which is present in the skin, right, which is present in the skin as affected, right? That means in skin also, right, in the skin, if I show you the section of the skin, you have got, this is epidermis, this is dermis, right? In epidermis you have got Langerhans cells. In the dermis also you have got lymphatic T cells, and now these T cells has affected, right? If these T cells which are present in the dermis has affected, of course there is a cutaneous problem, so cutaneous lymphoma. In skin, you have epidermis and the dermis, right? When if only one cell is present, malignant cell, okay, malignant cell predilection for, right? So what do you mean by epidermotropism? Right, epidermotropism is nothing but turning off. So why it is known as tropism? Why they have given the word tropism? Because this cell, single malignant cell, is turning off. That means from the normal it's becoming abnormal. So if there is one cell, you got an epidermotropism. Later there is collection of cells, collection of tumor cells, and now it becomes pus, abscess, and we call it as Poutry's microabscess. It is collection in epidermis. But in epidermis, you have got a malignant cells. Proliferate for epidermis in malignant cells, there is epidermotropism. It is known as epidermotropism, right? Because due to some stimulus in the T cells, now it is the normal cell is transferring to the malignant cells. So it is known as epidermotropism. Now if these malignant cells collect in the epidermis and it forms an abscess known as Poutry's microabscess. Right. So that's all regarding this mycosis fungoides. I told it affects the T cells. Pass positive, it is periodic acid is positive, and you can see cutaneous lymphoma. Right. I told you because it's affecting the skin. Now let's see what is Sézary syndrome. Right. Sézary syndrome is, now what happens here is, it is a complication or the late stage of mycosis fungoides. Now we have got a tumor cells in the epidermis. Now right now below you have got blood vessels. Now through the blood vessels, these tumor cells enter into the blood vessels and they spread, right? And they spread. So there is a cutaneous involvement before they spread. So it goes into the peripheral blood involvement. It also goes to it affects even the bone marrow also. So when I take the section, when I see the tumor cells, it looks convoluted, the nucleus looks convoluted, and we call them as cerebriform cells in peripheral blood. So that is all regarding the first session. We spoke about the mycosis fungoides. We spoke about the adult T cell leukemia. So just revise it once again by putting the DAMS notes beside. So let's meet in the next session. Thank you guys.